I would like to thank the organizers for giving me an opportunity to share some of our research with you. Today, I will talk to you about deploying protein missiles to take down disease-causing proteins from cells. But first, I would like to tell you about my journey to Dundee. I was born and raised in a remote village called Kimdanda in central Nepal. When I was 9 years old, I got a scholarship to study in a national school in Kathmandu, from where I completed my high school education. In 1995, for my undergraduate studies, I went to Bath University, where I studied biochemistry. Then, in 1999, I moved north to Dundee to undertake my PhD in biochemistry with Professor Dario Alessi. In 2003, for my postdoctoral training, I went to Memorial Sloan Kettering Cancer Center in New York. Then, in 2008, I returned to Dundee to set up my own lab at the MRC Protein Phosphorylation and Ubiquitination Unit at the School of Life Sciences, where I have been ever since. Talking about my experience of a very global training, since I started my research lab in Dundee, my research team of PhD students and postdoctoral fellows has been represented by 13 nationalities. This is the current team in my lab, and the picture was taken at a recent lab retreat we had in Aberfoyle. Cells are the building blocks of all living organisms, and trillions of cells come together to make up healthy, thriving human body. In the Sapkota lab, we are interested in understanding how cells communicate and respond to external as well as internal signals, and how miscommunication within and between cells causes diseases, and hopefully apply the knowledge to devise new ways of intervening against diseases. If you dig deeper into cells, roughly 20,000 proteins are responsible for executing every cell function and fate in organisms in response to a variety of external and internal signals. Therefore, protein function and abundance is tightly controlled to ensure healthy and functioning cells and tissues. Often, abnormal protein function through mutations, mislocalization, or misfolding results in diseases. Vast majority of approved drugs against diseases actually target specific protein functions in cells, typically with small molecule inhibitors. However, there is a great need for alternative strategies to target proteins. And in this context, we asked, could we target the destruction of disease-causing proteins? To try and address that, we have developed a technology that we termed Affinity-directed PROtein Missile system, or AdPROM for short. The AdPROM system is a multi-modular system that can be genetically encoded and packaged in retroviruses to deliver in desired cells. Like all missiles, at its core, it has two key components. First, the targeting module, which has to recognize specific target proteins. Second, the payload, which basically contains a destruction signal to destroy the target protein. First, for the targeting module, we use highly selective protein binders, such as nanobodies or monobodies. So what are these? These beautiful alpacas have been crucial to giving us clues about our target protein binders. Unlike humans, alpacas generate heavy chain only antibodies against pathogens. We can clip these antibodies and extract out the variable domain of the antibody that is sufficient for recognizing its target. These are called nanobodies, and relatively speaking, they are very small. They can be deployed to any cells, and they bind their targets with high affinity and selectively. Now, using the information from these alpacas, we can actually chemically synthesize nanobodies against potentially any target proteins in humans. For the destruction signal in the payload component of the AdPROM, we use enzymes called E3 ligases that naturally mark proteins for destructions. Any unwanted protein in cells is marked with ubiquitin chains by enzymes called ubiquitin E3 ligases. When any protein is marked in this way, they get destroyed by the proteasome, which is the cell's own waste disposal machinery. This actually has a lot of similarity with the City Council's waste management systems. Anything you put in the respective bins here, regardless of whether it is waste or not, it will get picked up and disposed of by the council. Now, if we deploy the protein destruction missile in cells, we should expect that the targeting module will go and bind to the target protein. Then the payload, which has the E3 ligase, will start marking the target protein with ubiquitin chains. This should trigger the destruction of the target protein by the cell's own waste disposal machinery. Right, time to test this against real proteins in cells. To do this, we collaborated with GSK to see if we could use our AdPROM system to destroy the K-RAS protein. RAS proteins are prominent oncogenes, and K-RAS is considered the holy grail of cancer targets because almost 25% of all solid cancers harbor K-RAS mutations. Although this has been known for more than 40 years, no approved drugs yet exist to treat these tumors. Therefore, RAS proteins are thought to be undruggable. We asked, could K-RAS be destroyed by AdPROM-mediated degradation? So we launched our K-RAS-directed missiles into A549 lung adenocarcinoma cell lines. And we find that indeed we can target K-RAS and HRAS for destruction. The question is, what about other targets? We next decided to target alpha-synuclein. Alpha-synuclein is a protein which aggregates in Parkinson's disease, and mutations in alpha-synuclein cause early onset Parkinson's disease. There are no treatments against Parkinson's disease available, and again we asked, could we target alpha-synuclein for destruction with our AdPROM system? Indeed, in 2 different cell lines, when we launch the alpha-synuclein targeting missile, we see complete degradation of alpha-synuclein protein. Based on these findings, we are now collaborating with Janssen Pharmaceuticals to test whether there are benefits to targeting destruction of alpha-synuclein protein in mouse models of Parkinson's disease. Finally, the AdPROM system that we have developed in our lab is a great platform for discovery. To understand the function of potentially any protein in cells, and a great tool for drug discovery, as well as if we switch the payload to allow other protein modifications, we can potentially uncover new drug strategies. Everything we do is an enormous team effort. Therefore, I would like to acknowledge my present and past lab members, our collaborators and support teams. I would also like to acknowledge the MRC, Janssen, GSK, Merck Serono, Boehringer Ingelheim, the Cunningham Trust, East Bio, and Michael J. Fox Foundation for funding our research. And thank you for listening.